Hello and welcome to our today's lecture of Introduction to Mechanobiology. In the last few lectures, we discussed about various diseases including cancer, atherosclerosis and muscular dystrophy. Apart from these diseases, we also discussed some basic cellular processes like cell migration, cell spreading and differentiation. So, processes like differentiation or any of these diseases take much more longer time to manifest, while cell migration and cell spreading requires the order of, so cells migration might have a cell speed of 10 to 100 microns per hour. So, for these processes you require that the molecules which participate are right there at the proper location, so that they can enact what they are supposed to do. However, for processes like differentiation, this is weeks in time. So, there are a lot of changes which are associated for a stem cell to differentiate into a neuron or a muscle cell or a osteoblast. So, what dictates it requires that these physical signals which are encoded by ECM in the form of topography or stiffness reaches the cell nucleus and activates various genes, and it is not just genes, you also have epigenetic changes. So, it is important to understand how from a cell periphery to the cell nucleus you have a signal which is transmitted and eventually activates a given gene. So, in that regard the nucleus becomes an important command center. So, if you think of the nucleus, so over the next 3-4 lectures we will discuss about the nucleus, what are its size, shape, composition and properties, physical properties like stiffness, and how do molecular players dictate these properties, and how are these properties relevant to processes like migration to differentiation. So, we begin with discussing the nucleus in general. So, as you all know that in contrast to cells like bacteria, in a cell the genetic information is stored inside the nucleus. And though we like to draw a picture, so our schematic of a cell with a nucleus might be like this, but if you look in side view, so if I draw a cell with a nucleus in side view, accounting for its proper shape and size, the nucleus will look something like this. So, clearly what you see is the nucleus is huge. And it is perhaps not surprised. So, it is of course the largest organelle inside the cell, and it is also the stiffest component. So, why is the nucleus the stiffest component? Think because you have the entire DNA. If you stretch out the DNA, you would have meters long, and this you put inside a nucleus with dimensions ranging from okay, order of 5 to 10 microns, okay, in size. So, when you compact such amount of DNA within the nucleus becomes very stiff. Okay. Now, what are the constituents of the nucleus? Okay. So, if I were to draw the nucleus, okay, if I were to draw the nucleus, you have actually friends. Okay. So, the nuclear membrane, okay, has two membranes. The nucleus has two membranes. The inner nuclear membrane or INM. So, this is your INM and this is your ONM or outer nuclear membrane. Okay. And within this you have the entire DNA which is present as chromosomes. Okay. So, some portion of the chromosome or the DNA is attached to the membrane, inner nuclear membrane. Okay. So, you have some portions where the DNA or chromatin, okay, is attached to the INM. Okay. And what I have drawn in red, these are nuclear pore complex. Okay. So, these are the holes through which proteins can enter and exit the nucleus. Okay. And typically proteins which enter the nucleus by themselves have what is called as a nuclear localization signal. Okay. And this entry is mediated by proteins like impotins, and similarly you have other proteins which mediate the exit. Okay. So, it is now emerging that there are hundreds to thousands of nuclear envelope proteins. Transmembrane proteins. Okay. So, these expression can vary across cell types. Okay. So, the question to ask, okay, is how do physical cues activate gene expression, okay, and can mechanically induced nuclear deformations control gene expression in a controllable, in a predictable manner. Okay. So, what are the molecular mechanisms that enable the nucleus to sense and respond to physical cues, and how properties of the nucleus are altered in various disease contexts. Okay. So, in this regard, okay, I would like to introduce this complex called a link. Okay. Link is the short form for linker of nucleoskeleton and cytoskeleton. Okay. So, inside the inner nuclear membrane, so if I draw a short segment, okay, of imagine that this is my outer nuclear membrane and this is my inner nuclear membrane. Okay. So, just underneath the inner nuclear membrane you have a network of intermediate filaments. So, this is a network of intermediate filaments. Okay. So, this is 25 to 50 nanometer thick, okay, and these intermediate filaments are called lamins. Okay. So, lamins are associated with providing structural support to the nucleus. Okay. So, while inside you have lamins, okay, so this is only one way connection. Now, as this term says, linker of nucleoskeleton and cytoskeleton, that means that there must be proteins. So, imagine you have this network of microtubules or actin. So, there must be some connections which exist which connect this. So, then you have a physical network from outside. So, this is your entire thing is your cell and this is just a short section of the nuclear membrane. Okay. So, this might be your actin stress fiber. Okay. Similarly, this one this will be microtubule. So, these molecules, okay, which connect the nuclear membrane to either the actin or the microtubule cytoskeleton are called nesprins. Okay. So, you have different type of nesprins. So, you might have nesprins 1, 2 which are known to connect to the actin cytoskeleton. Okay. You can have nesprin 3 which connects to the intermediate cytoskeleton. So, by intermediate cytoskeleton I mean vimentin network, okay, and you have nesprin 4, okay, which connects to the microtubule cytoskeleton, okay, via kinesin 1. Okay. So, if I draw this in a, if I draw the membrane in a little more detail, okay. So, let us say this is the, if you have these nesprin molecules, okay, they are known to bind to some proteins called sun proteins. Okay. You have sun proteins which link the nesprins, okay, to the nuclear membrane. Okay. And the sun in turn, so you have sun 1 or sun 2 and these in turn associate, okay, with the inner nuclear membrane through the cytoskeleton of the nuclear lamina. Okay. So, lamins they bind to large other nuclear membrane proteins, okay, and that is where you have other proteins like emerin or MAN1 as well as heterochromatin. Okay. So, lamins are known to bind to all of these. Now, lamins, given their structural role, they are associated, so they actually dictate the biophysical properties of the nucleus. Okay, of the nucleus, and it is envisioned that these lamins are envisioned to act as force transducers. Okay, in the nuclear envelope. Okay, and this is key to processes like nuclear anchorage, cell migration, or chromosome movements. So, there are various mechanisms which have been proposed as to how forces transmitted to the nucleus might lead to a signaling cascade controlling gene expression. Okay. One is like we saw for ECM proteins like fibrotin, you might have protein unfolding as one of the mechanisms. Second mechanism, you might have tension dependent stabilization of the lamina of the lamin network. And in some cases you might have chromatin stretching, thereby exposing spaces for binding. Okay. So, if you look at lamin expression, okay, so there are two types of lamins. Okay. In you have lamin A slash C. Okay. So, this is because of alternative splicing of lamin A gene, and you have lamin B1 and B2. So, you have A type lamins and B type lamins. Okay. So, most differentiated cells express lamin AC. Okay. And in stem cells, for example, in embryonic stem cells, lamin AC is very low, negligible. So, it has been demonstrated that lamin B type lamins, okay, are expressed during development. Okay. Now, you know that within the body you have this entire tissue stiffness scale, right. So, brain being hundreds of pascals, okay, to muscle order 10 kPa, to bone, collagenous bone order 100 kPa. So, in a seminal work, Dennis Disher and colleagues showed that, okay, so if you have an axis of tissue elasticity, okay, versus lamin A is to lamin B stoichiometry, okay, so you would find, okay, a linear relationship. Okay, so, let us say this is 1. Okay. So, below 1 you have in soft tissues. So, let us say this is, okay, 1 kPa order brain. So, this is your brain elasticity. So, you have cells. So, in this zone you will have neuronal cells. Okay. In somewhere here, okay, you would have liver, kidney, lung, and high here. So, then in this cluster you will have muscle cells, and high up you will have bone tissue like femur or skull. Okay. So, in other words, in soft tissues your lamin A to B ratio is less than 1, which means lamin B is higher, lamin A ratio is very less. Okay. While in stiff tissues this keeps on increasing. So, there is a increase in lamin A expression. Now, Along this, if you also plot how collagen levels scale with tissue stiffness, okay, so you would get a scaling relationship of collagen 1 scaling as bulk tissue stiffness to the power 1.5. Okay. So, this is called a power law relationship. Okay. Similarly, so you have this Lamin ratio, okay. Lamin A ratio scales as e to the power 0.7 and Lamin A to B ratio scales as e to the power 0.6. So, here E corresponds to tissue microelasticity. So, what it tells you is that as you increase in stiffness, so increase in ECM stiffness or increase in tissue stiffness is linked to increase in Lamin A to B ratio, okay, and increased Lamin A to B ratio is associated with increase in nuclear stiffness. So, this shows you that if a cell resides in an environment which is stiff, so it is likely that the forces on it is higher, it is subjected to more compressive forces or higher amount of tensile forces, and under those conditions the nucleus responds by increasing its expression of Lamin A compared to Lamin B, and the nucleus stiffens. Okay. So, this work we will, as reading assignment, I recommend you to read this paper. Okay. So, what in this study the authors also showed. Okay. So, when they look at the nuclear membrane, okay, if this, let us say if this is my Lamin A staining, AC staining, okay, and if this is my Lamin B staining, okay. So, if you co-localize, so there are segments in which they overlap, okay, there is complete overlap. So, suggesting that these two networks are juxtaposed. Okay. So, Lamin A/B networks are juxtaposed at the nuclear membrane. So, Lamin A is known to have multiple phosphorylation sites. So, including one at serine 22, one at serine 390, okay, one at, okay, this is a cysteine, okay, 522, okay, yeah, serine 390 and many others. Okay. So, what the author showed that if you do an experiment in which you take cells, okay, and you label all exposed cysteine residues with one dye, with a dye, okay, and then you subject the cells to shear stress. Okay. So, what force is known is going to lead to unfolding, okay, and this unfolding, so you have unfolding which leads to phosphorylation. Okay. So, what they have shown is if you track the labeling of cysteine 522, okay, the amount of labeling of cysteine 522 as a function of shear stress, you would see an increase in the amount of labeling, suggesting that as you expose forces onto the cell, these get transmitted to the nucleus, and as a consequence of this there is unfolding of this Lamin network, okay, and that is associated with increased amount of labeling of the cysteine reactive probe. Okay. Now, if you increase the shear stress at high shear stress zones, this actually the cells, this lamin undergo aggregation. Okay. So, this in these zones the cells actually undergo aggregation. Now, another interesting study was performed. Okay. In this study by Bucks, okay. So, what they did, they plated cells on soft gels where nuclei, where the cell is rounded and the nuclei is also rounded, and on stiff gels, okay, where the cell spreads and the nuclei is expected to be under lot more stress. Okay. And what they found, that there was an inverse relationship between phosphorylation of serine 22 as a function of stiffness. Okay. So, as you increase stiffness, phosphorylation decreases. On soft substrates, which means that amount of lamin phosphorylation is lot higher. An increase in lamin phosphorylation is associated. So, when lamin gets phosphorylated, okay, it leads to degradation. And degradation would mean that it would lead to effective nuclear softening. Okay. So, this is one of the mechanisms by which, depending on a soft or a stiff substrate, the extent of phosphorylation changes, and as a consequence the nuclear stiffness can also change. Okay. On a stiff surface, when the cell is exposed to lot more stresses, internal stresses, also you have stress fibers pushing onto the nucleus. In that case, the phosphorylation decreases and there is higher levels of effective lamin, okay, which contributes to the stiffness of the nucleus. Okay. With that, I stop here for today. Okay. In the next class, I will connect these observations of lamin phosphorylation and lamin expression to how these affect cell migration. Thank you for your attention.